Good morning, respected principal sir, our distinguished speaker of the day, Dr. P. Suryanarayana sir, PhD, postdoctorate fellowship, Lusanian State University, USA, and dear all participants. A very warm good morning. I am Harika, Assistant Professor in Department of Pharmaceutics in VVIPS and convener of this faculty development program. It gives me an immense pleasure to welcome you all to Day 6 of 1 Week International Online Faculty Development Program on cutting edge perspectives in pharmacy, translating molecules into
ये कितनी
medicines.
भोली वाली है ना। प्रथम में वो जो एफडीपी दे रही है उसका शक्ल कितना भोला भाला है।
This faculty development program has been remarkable platform for academic exchange, connecting global experts and participants to discuss the latest advancement in pharmaceutical sciences. Over the past few days, we have gained invaluable knowledge from distinguished speakers, and today's section continues this enriching journey. Now, now I kindly invite P. Bhagyareka ma'am to formally introduce today's distinguished speaker, Dr. P. Suryanarayana sir. Ma'am, please.
Good morning to everyone. It's my privilege to introduce Dr. P. Suryanarayana. Dr. P. Suryanarayana is a pharmaceutical scientist with expertise in drug delivery, pharmaceutics, nanomedicines, and tissue regeneration. He completed his B Pharm from Chebrolu Hanmaya Institute of Pharmaceutical Sciences, Guntur, M Pharm from Naipur, Mohali, and PhD from Naipur, Ahmedabad, where his research focused on advanced wound healing strategies using nanomaterials and smart drug delivery systems. He briefly served as assistant professor at various colleges, Shirpur, and gained industrial experience as a senior research associate at Sijin International Limited, Bangalore. Dr. P. Suryanarayana has contributed over 40 publications in peer reviewed journals, including ACS Applied Biomaterials and International Journal of Biological Macromolecules and ACS Omega and Molecular Pharmaceutics. His work spans the development of silver nanomics and optolaser responsive nanogels and liposomal drug delivery and exosomes based therapeutics strategies. He has presented his research at national and international conferences, and he is an active peer reviewer for journals such as PMC Pharmacology and Toxicology, Journal of Nanobiotechnology, and Quarantines in Medicine. His awarded awards include the ICMR SRF Grant, Best Poster Award at Biomedical Industry Research Day, and a travel grant to attend the IBRO APRC Associate School on Blood Blood Brain Barrier, and academically he He secured all Indian ranks of 11 in Naipur PhD entrance and 20 in Naipur Masters and 625 in GPAD. He is a member of American Heart Association and continues to advance research in nanomedicines and neuroscience and redox biology and tissue regeneration. Thank you, sir, for accepting our invitation. Over to Harika Madam.
Thank you, Ms. P. Bhagyareka, madam. Now I request Dr. P. Suryanarayana sir to take over this section and enlighten the participants with his valuable insights. Sir, please.
Yeah. Is my screen visible to everyone?
Yes, sir. It is visible.
Yeah. Thank you so much for giving this opportunity and inviting me for sharing my experience with all the faculties. And today in the today's talk, we are going to discuss the cannabinoids as a novel therapeutics for the neurodegenerative diseases. So the talk will go in a sequential manner. Initially, I will introduce what are the endocannabinoid systems and what kind of receptors where play a major role in the endocannabinoid system and how we can use them as a therapeutics for the neurodegenerative diseases. So let's understand what is endocannabinoids. Endocannabinoid systems or ECS is a complex system which is found in each and every cell of our human body. And what they are important for? They are important for maintaining in and crucial and important functions such as they will decide the mood, how we will behave, and they will decide when we have to feel the hunger, and they will treat us from the pain, and they will be involved to regulate how we sleep. So in a nutshell, these endocannabinoid systems are very important to decide day-to-day life. And what are the components of endocannabinoid system? There are three different components existed in this endocannabinoid system. The first one is endocannabinoids, that is cannabinoids which are produced by the body. We call it as endocannabinoids because it produced inside. That's why it is called as endo. And then second one is cannabinoid receptors. What they are proteins found on the cell surface that interacts with the endocannabinoids that body produces. And the last one is enzymes that breaks down the cannabinoids. Why that is important? Because anything excess production is not a good thing. So if the body produces high level of endocannabinoids, then that is also not good. So that's why to regulate that enzymes, such certain enzymes are there in our body, so that will break down the high excess amount of endocannabinoids so that they can maintain the physiological concentrations of endocannabinoids. So next, we will be looking what are the different endocannabinoids that body produces and what are the different endocannabinoids or phytocannabinoids we will get from the plants or natural resources. So the endocannabinoids like anandamide, which is called ananda, is came from Sanskrit, which is happiness. So when endocannabinoid anandamide was produced and it binds to the receptors, it gives us happiness. So that's why it is called anandamide. And then second one is 2-arachidonol like glycerol. These both were produced by our body system, whereas some of the natural constituents were there that mimics the functions of endocannabinoids. Those are called as phytocannabinoids if we are extracting them from the plant resources. Those are some of the examples of phytocannabinoids is tetrahydrocannabinol, cannabinol, cannabidiol, cannabigerol, cannabichrome. So these are the some of the examples which are produced by the plants, which gives same effects as endocannabinoids. And the third one is enzymes that breaks down the cannabinoids. Sorry, the second system is cannabinoid receptors that is presented on the cell surface. So there are two different types of cannabinoid system which is presented in our body. That is CB1 receptors, that is cannabinoid 1 receptors, and cannabinoid 2 receptors. So if we see In both males and females, the dots represents the cannabinoids. The dot which is in blue color is cannabinoid 1 receptors distribution in our human body, whereas dot CB2 which is pink color which represents the CB2 receptor distribution in our body system. So if we see, CB1 receptors are highly expressed in the central nervous system, whereas CB2 receptors are highly expressed in the immune system, that is majorly on the spleen and all the other immune cells which are presented in our body. So let's see the third part of the endocannabinoid system, that is enzymes that breaks down the cannabinoids. So here there are two different types of enzymes, that is fatty acid metabolizing fatty acid amide hydrolase that metabolizes the endocannabinoids. Another one is monoacyl glycerol lipase that will also involve in metabolizing the endocannabinoids which is produced in the body. So let's move from here. My entire story will be discussing more of CB2 receptors as a novel therapeutic target for treating the neurodegenerative diseases. Now, I have earlier said we have two different cannabinoid receptors, CB1 and CB2 receptors. So why we choose CB2 receptors instead of CB1 receptors, despite of CB1 receptors high localization in the brain. So if we see the CB1 receptors, the CB1 receptors is a psychoactive, it produces psychoactive effects despite of its high abundance in the brain. So this is a problem. Why? Because whenever we give a drug and if it goes and binds to the CB1 receptors, it activates all the receptors which are presented in different parts of the brain. And some of the receptors which are presented in the brain which will show psychoactive effects, like that will change the mood, that will change the heart function, that will change the brain function. So those are called as side effects in our like pharmacy. Whereas CB2 receptors, they specifically express only on the immune cells. So that gives a special effect, like specifically we can able to tune the inflammation by targeting CB2 receptors because they present only on the immune cells, not in the entire brain. So that gives us an advantage that targeting CB2 receptors will not produce any psychoactive effects. So that's why we choose CB2 receptors instead of CB1 receptor. And then there is a huge debate going in the scientific fraternity regarding the CB1 and CB2 receptor expression. CB1, everyone appreciated that CB1 receptors are highly presented in the brain, but few researchers said discusses that CB2 receptors doesn't present on the brain, and some of the research scientists they have showed that CB2 receptors are presented in the brain. So let's discuss whether the CB2 receptors are presented in the brain or not. So one of the research team recently have performed by extracting different parts of the brain, and then they have studied the CB2 receptors in different parts of the brain, whether that is expressing or not. And they have found, when they have done the analysis, they have found each and every different part of the brain, like olfactory bulb, neocortex, striatum, hippocampus, cerebellum, each and every part of the brain does show CB2 receptor expression. What it means? It means it confirms that the CB2 receptors are present in our brain system also, apart from the peripheral system. So now the second question is, okay, if it is present in the brain, what cells are predominantly expressing the CB2 receptors? Whether all the cell types which are presented in the brain were predominantly expressing the CB2 receptors, or are there any specific cell types which expresses this CB2 receptors predominantly? Now, when they have done the studies on the different cell types, they found out that on the microglia, that is a resident immune macrophages in the brain. So on the microglia, the CB2 receptors were highly expressed compared to the astrocytes, neurons, and endothelium. So it confirms that the CB2 receptors are present in the brain and which are highly expressed by the microglia cells. And then they have studied whether the child's and adult they express different concentrations of CB2 receptors or they express same level of CB2 receptors. So when they have studied in the hippocampus adult and hippocampus post 10 days of post birth, and they have in the mice, they have seen there is no significant difference between the CB2 receptors. The same results have been observed in the neocortex samples also. Adult, there is no significant difference in the CB2 receptor expression in the neocortex of adult versus 10 days of mice. And then further, they have checked in the nuclei when they have like specifically tagged with the CB2 receptor positive cells, and they found out even with the single nuclei FACS analysis, the microglia is highly expressing the CB2 receptors, but not other cell types in the brain. So what it gives the idea to us is microglia is resident immune cells, so which is involved in the tuning of or regulating of inflammation in our brain system. So one of so we have used this CB2 receptor as a target to treat the neurodegenerative diseases. So now what neurodegenerative disease we need to target? So when we have seen we focused on targeting the Alzheimer's disease. Why? Let's see what is Alzheimer's and understand the basic concepts of Alzheimer's. The Alzheimer's disease is a progressive brain disease that slowly destroys the memory and thinking skills by killing these cells progressively. And among the different forms of dementia, Alzheimer's is the most common form of dementia that contributes around 60 to 80% of all the dementia cases around the globe. And about 7.2 million people in the United States age 65 or older currently are living with the Alzheimer's, and more than 55 million people in the world are living with the dementia, and these numbers are estimated to be doubled by 2050. And though the exact mechanisms underlying these Alzheimer's pathology was not yet fully understood, around the globe, all the researchers in the Alzheimer's field were convinced that two pathological hallmarkers which are causing majorly the Alzheimer's, that is intracellular tau tangles and the A beta fibrils, which are extracellular A beta fibrils. So those were convincingly appeared in the Alzheimer's patients. So, and what are the different types of Alzheimer's? There are two different types of Alzheimer's, that is called as familial Alzheimer's disease and then sporadic Alzheimer's disease. What is familial Alzheimer's disease? Because these comes from our family hereditary disease due to genetic mutations. What are they? The genetic mutations in the APP, presenile and presenile-2 proteins. When the mutations occurs in these genes, these familial Alzheimer's disease occurs. Whereas the sporadic disease is more of lifestyle and aging and environmental effects. Those causes the sporadic Alzheimer's disease. Though the causes of both of them are very different, but they both share the common pathology, that is once they mature, the amyloid beta plaques forms and neurofibrillary tangles forms that shows the proteotoxicity, which subsequently lead to the neuronal damage, and then ultimately the cognitive impairment and memory loss occurs in the patients. So are there any drugs to treat this dementia or Alzheimer's disease? Yes, there are different drugs which are approved by the FDA. So since 1996 to 2014, the conventional drug deliveries were approved, which majorly aims to treat from the mild dementia to severe dementia. Means once the dementia is more severe or milder, from there they can able to manage those symptoms. However, recently from 2023 onwards, the MABs, that is the monoclonal antibodies, were approved for treating the Alzheimer's disease. So which can able to like cure the disease from the mild cognitive impairment. Means at start of the initiation of the cognitive impairment, these drugs can be given and slow down the progression of the cognitive impairment. However, one antibody recently which were approved, that is, say for example 2023, it means that post-marketing surveillance data of these MABs were scarce. Means we don't know whether these are very effective without showing any side effects, whether it is working in patients or not. One such example is aducanumab. So it was the drug which was approved by the FDA in 2021, but again discontinued in 2024. Why? So if you see the right side, this is the PET imaging. In the baseline, this is the disease humans PET image of the brains. Whereas after 1 year of dose administration of aducanumab, it can able to effectively reduce the A beta plaques. A beta plaques, the red color represents the A beta plaques. So if the red color is reducing, it means that it can able to effectively reduce the A beta plaques. So despite of its effectiveness in reducing the A beta plaques, it was withdrawn from the market due to the side effects which were observed in the post-marketing surveillance, which is like failed to demonstrate the significant benefit in slowing the cognitive decline. So even though they are clearing the A beta plaques, they are not helpful in improving the cognition. That was one of the reason it was withdrawn from the market. And the second one is it shows cerebral edema and intracranial hemorrhage when they have taken this aducanumab. So what these post-marketing surveillance data shows? So even the antibodies which are approved by the market, they were not still safe. They were not proven safe in the human post-marketing surveillance. Thus, we need novel targets to treat the Alzheimer's disease. That is one of the critical area, as there is no cure we have found it. So one such common pathway in the Alzheimer's is the inflammation. So even if it connects to the 10 different targets, ultimately in the Alzheimer's disease, the end stage of the Alzheimer's pathology is the inflammation, chronic inflammation. So if we can able to manage that inflammation, we may able to manage the symptoms of Alzheimer's disease. That's what the thought. And with that, we have targeted the CB2 receptors because these are highly abundantly expressed in the microglia, and the microglia is innate resident immune cells of the brain, and that plays important role in the inflammation. So can we able to tune the microglial function by using CB2 receptor agonist, so that we can able to manage the Alzheimer's disease? That was the thought. So with that, we have initially studied and find out that in the healthy human, the microglia expresses the CB2 receptors. However, under the pathological conditions such as Alzheimer's disease, the microglia expresses more CB2 receptors than the healthy receptors to manage the inflammation. Because whenever CB2 receptor expresses and it binds to the agonist, it changes the microglia from M1 phenotype to M2 phenotype. What is M1 phenotype? M1 phenotype is the state of microglia wherein the microglia produces more pro-inflammatory markers and shows the inflammation. Whereas whenever we provide the CB2 receptor agonist that binds to the CB2 receptors, and that changes the phenotype of microglia from M1 to M2, that is more of anti-inflammatory in nature, which shows opposite effects to the inflammation, so that it can able to down-regulate the inflammation which occurs in the Alzheimer's disease. So with that, So next thought was what drug we need to use. So we found out that cannabis is one plant. From that, there are 500 different types of compounds were extracted till date. There were much research happened on the cannabinoids. However, there weren't much research was focused on the terpenes which are isolated from the cannabinoids. So we focused on working on one of the sesquiterpene, that is caryophyllene, which is extracted from the cannabis sativa. And what are the why we have selected the beta caryophyllene? Because it is selective CB2 receptor agonist, which means it selectively binds to the CB2 receptors instead of CB1 receptors that is also presented in our human brain. And it is also naturally occurring compound which is extracted from the clavoid. And it is approved by the FDA, which means it is safe to administer. And it has demonstrated highly antioxidant, anti-inflammatory, and anti-cancerous actions. So that is the reason why we choose the beta caryophyllene as one of the therapeutic CB2 receptor agonists for treating the Alzheimer's disease. And then we have studied initially our study on the aging population. Why? Because aging is one of the risk factors for the Alzheimer's disease. So when we have done initially, we have done the cognitive functions between the young mice, that is 2 months old, and old mice, that is 11 months old. So when we have performed Y-maze, that is one of the cognitive function, we have found that aged mice was produced cognitive decline compared to the young mice. And then when we have administered the BCP, that is beta caryophyllene, at 100 mg and 178 mg in the old animals, which has produced cognitive benefits, which means it is enhancing the cognitive functions. However, if we are further increasing the dose, which does not show cognitive benefits. So that's why we choose 178 for further dosing and have seen how they are tuning the inflammation in the aging population. So when we have seen, interestingly, the markers such as IL-23, IL-27, and interferon beta were significantly downregulated when we administered beta caryophyllene to the aged population. So we tested. So here we have seen only in the aged population. Now we have taken this drug and we have tested this BCP for cognitive improvement in the Alzheimer's disease, whether they are producing any cognitive benefits in the Alzheimer's patients or Alzheimer's disease prone to mice. So we have taken BCP at 100 mg per case and treated the mice to check whether they have shown any cognitive benefits after injecting the BCP intraperitoneally at 100 mg per case. So here we have done the Y-maze test, wherein we place the animal in one arm that moves from this arm to means A to B and C. So if the mice moves, say for example, if I keep the mice in A and that subsequently moves to B and then moves to C, it means once the spontaneous alteration means their memory is good. But what if mice is moving A, B, and then again coming to A? It means that it could not able to remember where the animal has previously entered. So that means their cognitive abilities were lost. So in that way, we have done the cognitive test, and then spontaneous alterations we have seen in the wild type, that is healthy mice, and APP knock-in mice, that is one of the mutated mice which mimic the Alzheimer's disease mouse models. So in both in the wild type and APP vehicle, we haven't seen any significant improvement. I'm sorry. See, in both wild type and APP vehicle group, when we administer the 100 mg per case dose of BCP, in both of the genotypes, we haven't found any significant improvement in the cognitive function. However, when we have administered at 178 mg per mg, So we have seen significant enhancement in the memory function, means in the both wild types and APP mice also. So it shows that BCP is helpful in improving the cognitive functions in both wild type and APP mice. Means in Alzheimer's disease, it shows the cognitive improvement. And then further, same dose we have selected, that is BCP at 178 mg per case, we have taken and administered to the mice of the Alzheimer's disease model, that is APP knock-in mouse models, and seen the inflammatory profile such as IL-1, IL-6, TNF-alpha. These three are pro-inflammatory markers which are detrimental to any of the body if produced excess in our brain or even in peripheral body. So when in the APP knock-in, when it is untreated, the inflammatory markers were higher, whereas when we treated them with the BCP at 178 mg per case, all the inflammatory markers were goes down, which means the BCP is very effective in down regulating the inflammation. So with this, I conclude my talk. And if you have any questions, please guys, I'm happy to take.
Thank you, sir. Dear participants, if any queries, you are free to ask. Sir is there. Okay, I think so. There was no question, sir. You have explained in a nice, neat manner so that we can all understand it. So once you can stop sharing your screen, sir.
Yeah.
Thank you, Dr. P. Suryanarayana, sir, for your insightful and thought-provoking lecture. On behalf of BV Institute of Pharmaceutical Sciences, we express our sincere gratitude for sharing your expertise and valuable time with us. As a token of appreciation, we are going to present a certificate of recognition, sir.
Thank you
Sir,
so much.
please. Thank you for your support, sir. Though it was night there, you have supported us and making this an event grand success. Thank you, sir.
Thank you so much for everyone, especially VVIP Institute.
Thank you, sir. Thank you, sir. Sir, you can end the section.
Oh, thank you, man.
Okay. Okay, thank you, sir. We will send it through a mail, sir. The soft
Yeah.
copy. Thank
Sure.
you, sir.
Mhm.
And dear
Bye.
participants, thank you, sir. Dear participants, the attendance form, feedback assessment, and feedback form and today's assessment will be posted in the chat box. Please make sure to fill it out before leaving the section. So we have issued 3 Google Forms today. One is the 6th day attendance form. As usual, you are doing it from starting day 1 to day 5. Same as it. It will be a 6th day attendance link and one will be the feedback link. So feedback is must ensure. And the third one is assessment link. In it will be a questions of around 25. So everyone have to answer this. These questions are based on the international FDP which was conducted from day 1 to day 6. And who will get above 80%, they can receive an e-certificate. There is no time limit and these links are available till evening. So I request everyone participate to fill the Google Forms and submit in order to get an e-certificate. These 3 links are very important to get the e-certificate. If any of them are unable to fill, please kindly fill it again. And these links will be posted only in Teams. Just verify your chat box for the links. If any queries, you can ask. Okay. Then we are coming to the final section. Yes, we come to the conclusion of our 1 week international online faculty development program on cutting edges perspectives in pharmacy, translating molecules into medicine. It gives me an immense pleasure to express my heartfelt gratitude to all who have contributed to the grand success of this event. First and foremost, I would like to extend our sincere thanks to our respected principal, sir, Dr. A. Lakshman Rao Garu, for his constant support, guidance, and encouragement, which made this program a great success. His vision and motivation has been instrumental in organizing such academic initiatives. We express our deep gratitude to all our distinguished resource persons and keynote speakers for eminent institutions across the globe for sharing their invaluable insights on contemporary topics such as AI in drug discovery, nanotechnology, nanotherapeutics, and sustainable pharmaceutical practices. Your expert lectures have truly enriched our understandings and inspired meaningful discussions among the participants. A special note of appreciation to our FDP coordinator, Dr. Sheikh Aminabi Ma'am, Professor, Department of Pharmacology, for her dedicated efforts and coordinations and continuous support throughout the program. To all the participants, I thank you for your active engagement, enthusiasm, and thoughtful interactions during each sections. Your curiosity and participations made this FDP truly interactive and impactful. As we conclude, let us carry forward the knowledge, collaborations, and perspective guide through this program and continue to contribute towards innovation and excellence in pharmaceutical sciences. Once again, thank you everyone for your valuable contributions and participations. Now, let us conclude this enriching program with a national anthem.
There is no sound.
Dear participants, any questions?
Madam, yesterday, day before yesterday, link is not opening.
Sir, please.
Yesterday and day before yesterday, link is not opening for me. And there
Okay,
is a
sir,
Google
you
form.
please submit today and I will check.
Okay.
You are Srinath,
Oh,
right? You
yes,
have
madam.
messaged me in WhatsApp also, no, sir?
Yes, same,
Yes,
yes.
sir. Yes, sir. I will take care of
Everything,
that.
everything I'm trying filling it, but it is saying something went wrong
But it
at the
must
end.
say to fill the
No,
feedback link and assessment
no,
link.
I have filled and I have submitted, but it is not submitting, and the pop-up message is saying something went wrong
Okay, sir. Once you check
5
it.
times.
Okay, sir. Today feedback link and assessment link, there is no e-signature, sir. Just you have to choose the answers and you have to keep that's it, sir.
Okay, I will do it, madam. Thank you.
Okay, sir. Thank you. Any other queries?
Good morning, madam.
Yeah, good morning, madam.
Myself, Dr. Hari,
Yes,
from
ma'am.
Andhra Pradesh,
Okay,
MD
ma'am.
School of Pharmaceutical Sciences, Mohan Babu University.
Okay, ma'am.
The session is very interactive, madam. Actually, that for regarding pharmacology and docking studies. Those studies are very informative. We are thankful to our institute.
Thank you,
Our
ma'am. Thank you
institute is
so
nothing
much.
but pharmacy department. Pharmacy is nothing but our institute.
Yes, ma'am.
Okay. Thank you, madam. Thank you
Thank you,
very
ma'am.
much.
So the links will be open till today evening, and I'm going to verify the attendance and the assessment links, and I'm going to send your e-certificates within 10 days. And there will be a Diwali celebrations. No, after that we are going to check and we are going to send the e-certificates who are eligible to their respective email IDs. So kindly take some time. It will be I will send as early as possible within the time limit of 10 days. So after 10 days, if eligible candidate did not receive any certificate, please contact me. So thanks everyone. After filling the session, after filling the links, you can leave the section. Thank you everyone for your cooperation and making this an event a grand success. Thanks everyone for your active participations. Thank you all.
అంత ఫాస్ట్ గా అయిపోయింది. కాలేజ్కి వస్తేనే అయిపోతుంది. ఒక్కోసారి గడవదు. చాలా పనులు చేసాం కదా. ట్వంటీ 3 minutes ల్యాబ్ ఇప్పుడు మనం ఇంట్రొడక్షన్ ఏదో అనుకున్నామే, మనం మార్చి ఇది నీకు వాట్సాప్ పంపించేనా? ఇంట్రొడక్షన్ అదే నువ్వు కాపీ పేస్ట్ చేసి లేదంటే చాట్ జీపీటీలో అదే ఉంది. నువ్వు వాట్సాప్ ఓపెన్ అయ్యే ఉంది కదా, ఇందులో పేస్ట్ చేయాలా? ఇంట్రొడక్షన్ కాదు, ఇందులోనా? సెపరేట్ ఇంట్రొడక్షన్ లోనే పేస్ట్ క్లోజ్ పంపిస్తాను. ఇది ఎప్పుడు క్లోజ్ చేస్తున్నారా? నేను ట్యూస్డే కదా ఇస్తాను. ఆ వీక్ తీసుకుని చూద్దాం. ఫోన్ చేస్తుంటే ఆవిడ ఎత్తట్లేదు. మీకు పంపిస్తున్నానే. చూపేసి పంపిస్తుంది. ఇంకో ఇందులో అలా పెట్టు. నేను ఇందులో జర్నల్ చూసి జర్నల్ ఇది ఫ్రీ జర్నల్, చాలా మంచి జర్నల్. కాకపోతే అటు చూడు యూవీది యాక్సెప్ట్ చేసాడు. ఇదిగో డెవలప్మెంట్ అండ్ వాలిడేషన్ ఆఫ్ యూవీ స్పెక్ట్రోస్కోపీ యూజింగ్ మిథనాల్ అంట ఫర్ సైమల్టేనియస్ ఎస్టిమేషన్ ఆఫ్ డాక్సిసైక్లిన్ అండ్ వోరికనజోల్ యాంటీఫంగల్ pharmaceutical formulations. ఇది అయిపోయింది ప్రాజెక్ట్ ఆల్మోస్ట్ అయిపోయింది. ఇంకొక 2 కొన్ని అదేమైంది వీళ్ళు చేస్తారు ఈ ప్రాజెక్ట్ చేస్తారు ఇంతగా ఆర్టికల్ అంటావా ఏది చేద్దాం ఇష్టం నువ్వు చేపిస్తాను ఈ సంవత్సరం తర్వాత అది నెక్స్ట్ ఇయర్ పెట్టుకుందాం ఎందుకంటే ఇయర్ లో మనకి ఉన్నాయి కదా మళ్ళీ నెక్స్ట్ ఇయర్ దొరకకపోతే కష్టం అడిగారు నెక్స్ట్ ఇయర్ నేను అలాగా ట్రై చేస్తాను యాక్యురేట్ ఎస్టిమేషన్ ఏమనుంది చూడు ఇంట్రొడక్షన్ చదవనా pain associated with derivatives of total they are limited reports study aims to develop and elaborate the simple science. Okay. బానే ఉంది ఇంకోటి నీకు పంపిస్తాను అది కూడా యాడ్ చేస్తావా ఇది ఇప్పుడు క్విల్ బోట్ లో పెడదామా